Hi, and welcome to another episode of Christina Explains Stuff to You, specifically histology. So, this video is going to be the histology video for the integumentary system for my Biology 241 students for the first term of Human Anatomy and Physiology. So, before I get into the histology of it, I just wanted to give you a little overview about what you can expect to be looking at when you look at the histology of the skin. So your integumentary system consists of your epidermis and your dermis that cover the outer surface of your body. And so let's write that down. The epidermis is superficial to the dermis. And then below that is the hypodermis, which isn't technically part of the integumentary system. It's actually a connective tissue, but we talk about it when we talk about the integumentary system because there's not really any other time that's appropriate to discuss it. So this list goes from superficial to deep. So the epidermis is actually a specialized epithelium, and that is the keratinized stratified squamous epithelium. So keratinized refers to the fact that there is a hard, cornified, and dead outer layer of keratin, and that keratin comes from the keratinocytes that make up the epidermal layers below it. And this is why you're needing to exfoliate because your epidermal cells are always turning over. They're highly regenerative. Below that is the dermis, which is a mixture so there's a thin layer of loose areolar connective tissue right under the epidermis. We call it the papillary layer because it's making up the dermal papillae. Don't worry, if you don't know what I'm talking about, I will show you. And then below the papillary layer of the dermis, there's the reticular layer, which is dense irregular connective tissue. So as the name suggests, for dense irregular connective tissue, it is dense and the collagen fibers of it are arranged in irregular, non-parallel, sort of crisscrossing mixtures of fibers. And the dermis is where you're going to find the accessory structures of the integumentary system. So things like sebaceous glands, sweat glands, hair follicles, various corpuscles that allow detection of the touch sensation, etc. And the hypodermis is fat and other connective tissue that basically allows for two things. Insulation and nutrient storage, specifically triglycerides. So you have a subcutaneous fuel source as well as insulation, and it also allows for skin to slide over the underlying muscle so that your skin is not adhering to your muscle. So these are the kinds of things that we're going to look at and observe when we look at the histology of the integumentary system. So what you're going to notice, and I'm going to come in here and highlight, or let's highlight, why not? I'm going to highlight these in pink. What you'll notice is that as I'm describing the histology of the integumentary system, I am referencing some of the tissue types that you're supposed to learn along with the integumentary system histology. So this is the first time that you're going to have practiced this skill. The reason we make you learn the tissue types is because you will figure out what part of the body you're looking at based on your knowledge of what tissue types it is made of and how those tissue types come together to form the tissue and or organ. So here we have loose areolar and dense irregular connective tissue, and here we have fat and other kinds of connective tissue. So really, all of the histology of the integumentary system is a specialized set of the basic tissue types, specifically keratinized stratified squamous epithelium is kind of the big one, and then everything else is just different and various arrangements of other tissue types. So, having said that, let's go look at the actual histology. So, I'm gonna just undo my little whiteboard there. So, skin comes in two varieties. There is thick and thin skin. So as it says on histology guide here, thick skin is only found on the palms of the hand and the soles of the feet. So thick refers to the thickness of the stratum corneum specifically, which is that outer dead layer of cells. So it has a thick epidermis and contains sweat glands, but no hair follicles and no sebaceous glands. So imagine if you had oily, hairy palms, it would be awfully hard to grip anything, right? And also probably no one would want to hold your hand. So thick skin is specially adapted for gripping, and that's why we have friction ridges and this lack of hair follicles and sebaceous glands, because those would get in the way of gripping stuff. Thin skin is literally everything else. So everything except palm and soles. Now, the actual thickness of the skin varies over the surface of the body. And what I mean by that is even within thin skin, there are areas of it that are thicker than others. So for example, the skin of your back is thicker than the skin of your eyelids, for example. But it's still not considered thick skin because it doesn't have that really robust stratum corneum. So let's first go look at thick skin, and then we will look at thin skin, and we'll look at what kinds of things are found in each. So let's see, what do I want to do here? I want to do this one. So one thing that you're going to want to sort of avoid when you are studying histology is the idea of up and down. So you would probably expect the superficial layer of the epidermis to be quote unquote up, therefore like at the top here somewhere. However, orientation doesn't matter so much as being able to find the apical surface. And so for histology guide, this is a really nice piece of thick skin. I chose it for that reason because it has a lot of the features that I want to show you, but it's on its side. So just keep in mind the terms up and down are relative, and it's important to bear that in mind when you're analyzing and looking at histology. Now, I also mentioned edge finding. So epithelia are highly cellular and organized. So if you're looking at this picture and you're like, I'm going to find epithelium, I'm looking for keratinized stratified squamous epithelium, you're going to look for a highly cellular organized area. So not this side, but this is fitting the bill. So that's where I'm going to zoom in. And we're going to do some labeling. So first things first, let's label the apical surface. So that is the free edge of the epithelium. On the inside of you, the free or open edge of the epithelium is lining a lumen, but since this is our skin and it's covering our surface, it's just the outer layer of skin. So since this is thick skin, and I'm going to use the highlighter function here, what we are doing is looking at, this layer. So I'm going to highlight the layers in different colors and then I will label them. So just bear with me for one moment. Okay, so I made you a cute little rainbow. So now let's do the corresponding colors. So I'm going to make a little key over here. Actually, I'll make it up here so that we can see all of it because it's easier if we have some background colors. So the yellow layer is the inferior-most or most basal layer. So this yellow line I'm making here is the basement membrane. And the single layer of cells right above it, so just these cells here, so you can see the bottom layer of cells, is the stratum basal, also sometimes called the stratum germinativum. Let me write that end better so you can actually see. That's a V, this is a U, that's an M. All right, so next up, we've got a layer that's only present in thick skin. It's not present in thin, so for that reason, I'm going to put an asterisk here. And this is the stratum lucidum. So it's a very thin layer of sort of semi-transparent dying or dead cells right above this dark purple band here. So I'm bracketing the stratum lucidum here and here and here. So that's what it is. So it corresponds to the area that I've colored with green. The thickest layer, except for the outermost one, which we'll talk about in a minute, the one in orange here, is the stratum spinosum. So I actually have the orange blob in the stratum spinosum itself, so that's going to go from here to here. And then our little pink layer, oops, I dropped my pen. Which is this very dark band here, is the stratum granulosum. So that layer is the layer in which the keratinocytes are filling up with keratohyalin granules, which is why they have this exceedingly dark appearance. And they're in the process of dying because as they move up, the next thing they're going to become is our blue highlighting, which is the stratum corneum, which is this. So that's a cornified layer of dead keratinocytes, which have filled up with keratin and then died. So you'll notice that you don't see any nuclei here in this layer, and that's because the cells are dead, not alive. I'll do a little like dead smiley face here. So the presence of a nucleus indicates a living cell. So if you don't see any nuclei and you see a big layer of nucleus-free dead cells, that is your cue that that is the stratum corneum. So notice these are color-coded according to these stripes. So I'm just going to indicate these once again. And I didn't exactly do them in order. That's not important. What is important is that you are able to identify each layer. So the way that this works is in the stratum germinativum at the bottom, the majority of the cells, each individual one of these, are basal cells, which are a kind of stem cell. So when each one of these divides, it produces one daughter cell that stays a basal cell, stays a stem cell, and the other cell moves upward and becomes a keratinocyte. And then in the stratum spinosum, keratinocytes undergo lots and lots of mitosis, and you can even see some of that. So like this cell looks like it's undergoing mitosis, and probably this one as well. In fact, that one looks like it's in telophase. So this is where keratinocytes do lots of mitosis to divide and make more of themselves. So they are becoming keratinocytes, and then those keratinocytes are going to undergo division so that they can become very numerous. And then in the stratum granulosum, those keratinocytes are, I'll draw this up here. Are filling with keratin, and they are also undergoing apoptosis or programmed cell death. So they're supposed to die. It's not an accident. And then after that, the stratum lucidum and corneum are just made of non-living keratin-filled cells that function in a variety of ways. So among the functions of the stratum corneum, it is waterproof. It is shear resistant, meaning it's hard to tear or rip. And it is a barrier to pathogens. So this is why having a intact cutaneous membrane, which is another way to talk about the integumentary system and the epidermis, having an intact cutaneous membrane is a good way to keep nasty pathogens like bacteria, viruses, and parasites out. This is why those kinds of pathogens tend to like to get into you via cuts or abrasions or any sort of injury to your skin surface. So those are the layers. Now I'm going to delete this, and we're going to go look around the slide and see what else we can find. Okay. Ooh, that's a very good one. So what you're going to notice if you look carefully is that the interface between where the epidermis ends and the dermis begins is not flat. It's not a straight line. It is wavy, and that is what I'm outlining here. So each of these little finger-like projections of the dermis up into the epidermis is called a dermal papilla. So plural is dermal papillae. And papilla is actually Latin for nipple. So dermal papilla means skin nipple. So anything that's nipply shaped is called the something something papilla. So because these papillae are made of dermis, then they're called dermal papillae. So this is just a name for the specific shape of these extensions of the dermis that poke up into the epidermis. So the papillary layer of the dermis is the layer that includes the dermal papillae, and then everything below that is the reticular layer, as I mentioned previously. So within dermal papillae, you can sometimes find a very specific kind of touch sensory organ, and that is called a Meissner's or tactile corpuscle. So what you're going to notice is there's this sort of thumbprint-like clump of cells inside of this particular dermal papilla. And if you look at it, you'll notice that in the center of it, there are no cell nuclei, and the cell nuclei are kind of pushed off to the side. And it looks like a little thumbprint. So that is a Meissner's / tactile corpuscle. So Meissner is the guy who named it and found it. Tactile refers to its function. So you can either say Meissner's corpuscle or you can say tactile corpuscle on the lab practical. Those terms are interchangeable. Either one is fine. So that's pretty cool that you can see that, as well as the dermal papillae. And in order to learn to recognize Meissner's corpuscles, it's good to be able to visually look, which is why I'm going to delete this circle here, and just compare this particular dermal papilla with a Meissner's corpuscle in it, compare it to this one, for example. Let's circle that in different color, actually. I'm going to do blue. So this dermal papilla and this dermal papilla do not contain a Meissner's corpuscle. And you can see because it's just regular connective tissue, the nuclei are not arranged in any particular way, and it stains more lightly. This one does, and you can see that it's clearly different and has this thumbprint-shaped array of cells within which the nuclei of those cells are kind of pushed out towards the edge of the structure. They're not in the center. So Meissner's corpuscles are responsible for fine touch, vibration, and texture. So, for example, if I blindfold you and then I hand you microfiber in one hand and real silk in the other hand, you will be able to tell the difference between the two, even though one is a mimic of the other. And you'll be able to do that because you have these very sensitive little detection arrays in the form of Meissner's corpuscles in your fingers. So let's zoom out a little bit and just go have a look, see what else there is to behold. I don't see any other Meissner's corpuscles in this slide, just that one. I see lots of dermal papillae. So I see lots and lots of collagen fibers, all these pink things. In fact, most of this pink tissue in general is collagen. And that's because this whole area of the dermis is made of dense, irregular connective tissue. And although there aren't any like really spectacular sweat glands in this particular image, you can see a sweat gland duct, which is a little piece of cuboidal epithelium tube that you can see is kind of heading up towards the surface. But because of the way this was sliced, we've only captured a portion of the duct, not the entire thing. And there's another one of those down further, which I'll also show you before I switch slides. So here's a really good one. See that cuboidal epithelial tube climbing up towards the apical surface? That's going to release sweat onto the apical surface, both to cool you and also to create a thin film of water that allows you to have extra gripping capabilities. So now let's go check out another piece of thick skin. This one has much better sweat glands, but first we're just going to have a peek at the epidermis and the dermis. So this particular slide has a much more pronounced and easier to see stratum lucidum. So you see this like light translucent layer that divides the stratum corneum from the stratum granulosum. That is the stratum lucidum. And remember, this is only present in thick skin, so you're not going to see it in thin skin. So that's pretty nice that we can see that. We can still see dermal papillae. So all of these little extensions and projections. And then if we look down here in the dermis, because I don't really see any Meissner's corpuscles up here, I can once again see sweat gland ducts climbing up towards the apical surface. So let's zoom in on those and have a closer look, shall we? And in this case, you can even see the duct passing through the stratum corneum, which is neat. So if we go down further, we can kind of visually trace these ducts. So this one, for example, I can see this and this are in a line with each other, so they're probably part of the same sweat gland duct, and they're most likely associated with this set of sweat glands. So what you're going to see are little clusters of tubes made of cuboidal epithelium that are anchored in the dense irregular connective tissue of the dermis. So see all of these, we have little lumens, which are narrow, but they're there. And then surrounding those lumens are tubes made of stratified cuboidal or sometimes simple cuboidal epithelium. So each of these is not only a sweat gland, but specifically, I'm making myself a little space to write here that's a little bit easier to see. These are eccrine. Oops, I want that to be a little bit smaller. Slash merocrine sweat glands. So eccrine/merocrine sweat glands produce true sweat, which is the sweat that's for cooling you. They are different than apocrine sweat glands. Those are the sweat glands that are found in your groin and your armpit, and those sweat glands have an additional feature, which is pheromone secretion. But these guys are eccrine/merocrine, so they're going to produce true sweat, which is cooling in nature. So let's zoom out a little bit, and we're just going to go look at some other sweat glands that are in this field of view. So here's a big cluster of them. Here's a cluster of them. Here's a cluster of them. So you can see they're pretty numerous if you look at them and learn to look for them. And then you can see the ducts that lead away from them and are going to carry that sweat to the surface of the skin, to the apical surface of that keratinized stratified squamous epithelium. So I mentioned the hypodermis as well. Oh my. And in some cases, the hypodermis and the dermis end up mixing a little bit. So you can see this adipose tissue. There's quite a bit of it. And there's more and more as you go down. So these are fat cells. Another thing you can see really well in this particular piece of thick skin are Pacinian corpuscles. So what you're going to look at is see these arrays of cells that look kind of like a cut onion, where there's multiple concentric layers of cells. These are Pacinian corpuscles. And these are a special kind of touch sensor that are used to detect deep pressure and touch. So if someone gave you a firm handshake and you can feel the pressure of them squeezing your hand, that's going to be detected by Pacinian corpuscles. So these are basically just layered arrays of sensory cells that have specific ion channels in them that respond to being pushed on or poked. And they look like the cut face of an onion. So we'll see some other examples of these as we move through, but I wanted to point them out since they're here. You can also see cross sections of nerves. So these are nerve cross sections, these little round guys. And then down here, this is an artery for blood supply. So remember, sometimes I'm going to show you stuff on the slides for context that is not necessarily on your list. So artery is a good example of that. I don't expect you to recognize arteries yet, but since we have one in front of us, and since I don't want you to mistake one of the Pacinian or other corpuscles for an artery, I'm just pointing it out since it's in front of you so that you run less of a risk of mistaking it for something else or vice versa. So all in all, that was a really nice piece of skin. So let's go look at thin skin by contrast. And specifically, I'm going to pick the scalp because I want to look at thin skin that also has hair follicles. So what you can see right off the bat, if you look closely, is that the stratum corneum is much thinner. So unlike the big, huge stack of keratin that was sitting on top of the thick skin, the stratum corneum in thin skin is relatively thin and wispy. So that's a hallmark of thin skin, is the stratum corneum looks more like this. And then you can see all of the other layers are also small as well. They're just not as thick, each of them, as in thick skin. They're all still present with the exception of the stratum lucidum, but they're just narrower. So for that reason, I typically like to ask you about the epidermal layers on a thick skin slide rather than a thin skin one, just because it's easier to point them out and differentiate between the epidermal layers if I have nice thick layers to work with. Okay, so we did not come here for the epidermal layers. We came here for the accessory structures. So here you can see this is a hair. And if you follow it down, we can see its follicle. So let's do that. So each of these things, oops, I didn't want that to be black. There we go, is a hair follicle. Some of them you get a nice longitudinal section. Some of them, depending on the angle of the cut that produced this thin section, you only get a little piece of, like here. So these are all, let's make that a different color so you can see. Hair follicles. So if you look, what you're going to notice is that the epidermis doesn't stop. It actually continues and dips in and becomes the hair follicle. So you can actually follow it down. That's because the epidermis and the keratinocytes that make it up are what produce keratin. And hair is also keratin. It's just a modified form of it or different shape of it. So it makes sense that the epidermal layers are going to sort of fold inward to form the hair follicle. So it's just a specialized epidermal structure at the end of the day. So around each hair follicle, kind of like a skirt, you're going to see these sort of fan or roughly triangular or teardrop shaped clusters of cells. And I'm going to circle all of the ones I can see here. They're always going to be associated with the hair follicle. So these are sebaceous glands. So sebaceous glands produce sebum, which is an oily secretion, oil and dead cells. So this is the stuff that you can squeeze out of your pores if you're picking at your face. It lives in your hair follicles, but if it builds up in a pore, you can squeeze it out. Um, it's also the stuff that if a pore becomes blocked and infected, the reason that it becomes inflamed is because bacteria like P. acnes, which is the bacterium that causes acne, will begin to feast on the delicious and nutritious oily secretions, and that activates the immune system and causes inflammation. But sebaceous glands produce this oil for waterproofing and to keep skin supple. So it keeps your skin from drying out and cracking because it's oily and like most moisturizers, it's oil-based. We can also see sweat glands in this picture. So see these cuboidal epithelial clusters of tubes here, here, there. There's some more right here. So these are all sweat glands as well. And these are still eccrine or merocrine sweat glands because we are in the scalp. And remember, the apocrine sweat glands are only in your armpits and groin. So let's cruise around and see what else there is to see. There is one structure that I need to show you that I want to find a good example of. So I'm just going to kind of scan for a good example of the thing. Aha, there we go. Perfect. Okay, so let's color code this sucker so I can show you the different parts. So I can see my hair follicle in an oblique section, which means it's not quite longitudinal and it's not quite cross-sectional, it's diagonal in between. And then, as I promised, coming off of it in sort of teardrop or triangular shapes is the associated sebaceous gland. And then this is a bit of smooth muscle, and you'll notice that it attaches to the side of the hair shaft. So this bit of smooth muscle has a specific name, and that is the erector pili muscle. So this is smooth muscle that erects the hair. So when you get goosebumps on your arms, for example, when your hair stands on end, that is your erector pili muscles tensing up and pulling our hairs into an upright position. In a really extreme example of that, if you have a dog or you've seen a dog get upset and agitated and aggressive, and you've seen the hair on their back raise up, that is their erector pili muscles raising their back hair in an aggressive or threat display. So that's the muscle that's responsible for that. So erector means a muscle that causes something to become erect, and then pili means hair. So erector pili muscle is just the hair erector muscle. So let's go look at some other examples of the erector pili. I can see some streaks of one here and here. Let's go cruising for another piece. I can see one here. So you'll notice that depending on the slice and the angle of it, you're not going to always get a big obvious erector pili muscle like the first one I showed you. Some of them will be less conspicuous or less obvious, although this one's pretty nice. So you can see they kind of attach at an angle to the shaft of the hair follicle, and there's often, as is the case here too, they're often sort of cupping a sebaceous gland. So you see that? So let's go look at some other examples because having many examples is good. Oh, look at that. So there's an erector pili muscle, again, cupping a sebaceous gland. Here's a really nice long erector pili muscle, and you can see it's cupping a sebaceous gland. So there's this repeating pattern where you've got hair shaft and follicle, sebaceous gland attached to that, and then sandwiching the sebaceous gland in is the erector pili muscle. I also want to zoom in and show you that you can actually see, oop, histology guide's going to buffer a little bit. So here is a place where you can actually see that the sebaceous gland is opening onto the hair follicles. See that right there? So it's opening up here. So this pink stuff is going to be sebum. And, sebaceous glands are holocrine secretion, which means that actually the cells that make up the gland become the secretion. They die and fill up with oil and make the sebum. Also, I'm noticing something spooky here. This little piece of sebum looks like a creepy face where like, oops. There's like a mouth, a spooky skeleton mouth, and a nose, and some eyes, and little horns, and then a chin. Creepy. Let's give him a name. We're going to call him Spooky Stephen the Sebum Demon. That's just an example of human pattern recognition. Human brains are encoded to try and find patterns in our environment. So sometimes people see Jesus Christ on a piece of toast. And sometimes, you're just cruising around a microscope slide and you happen to run into Spooky Stephen, the sebum demon. Hello, Stephen. There you are. But really what that is just dead cells and oil. Sorry, Stephen. Okay, back to business as usual. So I'm just gonna kind of cruise around here and see if there's anything else interesting I can find to show you. But it's not looking like it. No. See some nerves, see some adipose tissue. Yeah, nothing really exciting happening down here, so I think it's time to move on to another slide. Goodbye, Stephen. So here's another view of scalp. This one has more cross-sectional hair follicles, so you can see there's circles instead of lines, but it does have a really nice hypodermis. So this big layer that's full of fat is the hypodermis. And you can see it's very distinct from the dermis, which is here. Very obviously different tissue because, again, adipocytes store triglycerides, and triglycerides do not like to pick up either hematoxylin or eosin, so they don't pick up stain very well, which is why the hypodermic fat appears stainless. Okay, so we've got Meissner and Pacinian corpuscles, and it looks like the same sets of slides as we had above. So let's go check this out. Oh yeah, I see some good ones. Okay, so both Meissner's and Pacinian corpuscles you will find in the dermis, but the Pacinian corpuscles are deep in the dermis, whereas the Meissner's corpuscles are superficial. So the reason I got excited and said ooh is because I saw a really nice Pacinian corpuscle, and it is this one. So you can see it looks like an onion. If you cut like a red onion in half, it looks like a cut onion. So this is a Pacinian corpuscle. And this is a sensory structure that allows, again, detection of deep pressure and vibration. So That's a Pacinian corpuscle. Very conspicuous, very obviously not a gland or a duct because there's no lumen. It's just this sort of multi-layered onion skin looking structure that's deep in the dermis. Let's see if we can see any more. Oh, there's a couple more down here. These ones aren't quite as round, but they meet the same criteria. So we can see one here, here, here. So again, onion skin appearance. Not necessarily perfectly round, although that first one I showed you was pretty round. But as you can see, same. So there's concentric, circular or oval layers of cells wrapping around each other. So we know that we find Meissner's corpuscles / tactile corpuscles. Those are found inside of dermal papillae. So if you're looking for one of those corpuscles, what you're going to want to do is just scan through the dermal papillae until you see something that looks like a thumbprint. Oh, like this. So there's one. You can see it's a little aggregate of cells, and within it, most of the cells' nuclei are pushed out towards the edge. So let's keep scanning. So what you're going to notice is that not every dermal papilla has a Meissner's corpuscle in it. Only some of them do. I'm going to pause for a moment here to point out this very excellent sweat gland duct, and you can even see it going up through the epidermis and the stratum corneum. But we're going to continue sort of trolling for Meissner's corpuscles. Oh, there's one. It's a little one, but it's still one right there. And let's keep looking. No, no, no. This is one, but it's probably not one I would pick for a test because it's not especially conspicuous. I like the when they look really thumbprinty and are very large and obvious, like this one is. That's a good one. And let's keep going and keep looking. None of these have any. Here's one. Again, not a great one, but it is one, so I'll show it to you. So you'll notice that they're not very frequent. They do exist, but they're not in every single dermal papilla. So you have to look for them. They're not going to be a guarantee. Ooh, this one here is really nice. So it looks like a thumbprint. This one is okay, but this one is a good one. So looks like a thumbprint sitting in the middle of a dermal papilla. And that's the end of this slide. Okay, so I have one more thing I want to show you before I conclude this video, and that is pigmented skin. So melanin is a pigment that is made by a specialized cell type called melanocytes. And then what the melanocytes do is they make this pigment, which is dark brownish black most of the time, although there are different kinds of melanin. And they pass that melanin up to the keratinocytes above them so that those keratinocytes can use the melanin as a shield from the sun. What melanin is a little molecular umbrella. So the keratinocytes put the melanin on the side of their nucleus that faces the sun, and that helps protect their nucleus from ionizing radiation. So melanocytes only live in the stratum germinativum. They're not in the stratum spinosum, and they sit among the basal cells. So most of the cells you see in the stratum germinativum are basal cells, like these. But then, let's make this brown because that seems appropriate, we've got these little guys, and you can see they have like a little white space next to them. This one's a really nice one. So is this. Those are melanocytes. So when you are scrolling through the stratum germinativum and you come across a cell with a little white space next to it, those are going to be melanocytes. So the stratum germinativum consists of basal cells interspersed with melanocytes. So let's just go continue to find another couple examples because it's always good to have multiple visual examples of stuff you're trying to look for. So here's another melanocyte, here's another melanocyte, here's one. So again, there's this consistent theme of having a little white space next to them. And there's a couple more. So this one, this one, this one, this one. So those are all melanocytes. Now, because this skin is stained with an additional dye, it's not especially easy to see specifically the melanin that is being used to protect the cell nuclei, but you can see the melanocytes quite well. This is a little bit darker, and you can see areas of darker pigmentation and some even leaking out into the stratum lucidum. Also, really nice stratum lucidum here. So this is what melanocytes look like in pigmented skin. Now, non-pigmented skin, white skin, also has melanocytes. It's just that those melanocytes are going to make melanin at lower rates, and the keratinocytes are not going to accept the melanin to the same extent. Okay, so that concludes our histology tutorial on the integumentary system, and that concludes the material that is for the first practical in Biology 241. So the next histology video I make is going to be a short quiz histology video where I'm going to give you some sample test questions and have you pause the video and try to answer on your own, so it's going to be a little bit interactive. Thank you for your attention, and I will see you in the next video.